Okay, so hello everyone. I'm Noemi. I'm one of the student committee members of the Global Renal Exercise Network. And today is our fourth webinar, trainee webinar, where we're going to sort of present some of our members' research. So just a little bit about us. We are the Global Renal Exercise Network. We are researchers, medical and allied healthcare professionals from around the world who aim to foster collaborative research and innovation across multiple disciplines. And one of GREX's or GREX's main objectives is to develop effective strategies to increase physical activity and optimize health outcomes in people living with a kidney condition. Therefore, to engage more researchers and professionals, GREX decided to initiate a GREX trainee webinar. So we all know that the current situation had a massive impact on all of our lives, and it also prevented us to present it at conferences this year. So we would like to give the opportunity for our members to present their work that they may have submitted to other conferences. The primary purpose of the webinar today, and generally our webinars, is to provide the more junior members of GREX to present their work. So that doesn't necessarily just mean students, it means research assistants, healthcare professionals, or postdoctorate or early career researchers, or technicians who are interested in this field of research. So previously we've had 3 successful webinars. The first one took place in May, and a month after that we had another one in June, and the last one was in July. So the reason I'm showing this to you is if you're interested in any of the titles or want to ask some questions, I'm sure the authors of these would be happy to answer them. And today, we've got 3 presenters who I'm going to introduce in a minute, talk about their research from the US, Australia and the UK. Just before we get started, each abstract is allowed to have 10 minutes, followed by 5 minutes of audience questions and answer. And if you have any questions, please do ask that in the chat, or if you have more specific questions related to one of the presentations today, then we have another like 15 minutes at the end where you can, ask the questions to the authors or discuss anything that you're interested in related to GREX or GREX. If you haven't done so already, please follow us on Twitter, or check our website to learn a little bit more about GREX. So I'm going to stop sharing my screen and let Brett, our first presenter, to get ready. And until he gets ready, I'm going to read a short bio of Brett. So Brett Burrows, he's a PhD student at the University of Illinois in the US, supervised by Dr. Ken Wieland. His research efforts include investigating the effects of exercise and nutrition-related interventions on the health and quality of life of hemodialysis patients. He's also investigating behavior change techniques to optimize the efficacy of these trials and to improve exercise and nutrition-related outcomes. So Brett, happy to get started.
All right, thank you so much, Naomi, for that introduction. Appreciate that. So today I'll be giving a talk on my research that we conducted about a year ago, right before COVID hit. So we were able to actually do it, which was great. But fully immersive virtual reality-based mindfulness intervention in hemodialysis patients, and this was just a pilot study assessing safety and utility. So just some background, virtual reality in general is being used more and emerging in technology in the medical field in various forms. Currently it's being used in the medical field in medical procedures and surgeries using full immersive VR. It's also being used as a psychotherapy tool and for pain management patients. And then there's also currently, or past previous trials been looked at in hemodialysis population using non-immersive VR. So 3 of the 4 trials have used a Nintendo Wii. So that picture there on the right-hand side with the individual where they're doing some type of movement, and that is displayed on a television or computer screen. But to date, there has been no trial exploring the potential benefits of fully immersive VR using goggles that go over your eyes, that is kind of represented in that top picture. So why are we possibly looking at fully immersive VR for hemodialysis patients? So about 3 years ago, we did a feasibility study for an internet-based positive psychology study to reduce depressive symptoms in hemodialysis patients, and we used Apple iPads to deliver the intervention, and we found some significant barriers through the program. So literacy concerns, holding the iPad during dialysis was an issue. So not only holding it, but then also navigating the website was an issue as well. And then internet disruptions. So if the internet went down in the clinic for the day, we could obviously not run the program for that day. So we thought possibly fully immersive VR may be able to either reduce or eliminate some of these barriers. So our study aim was, again, it's a pilot study, so we were just looking at the safety and accessibility and utility of using a fully immersive VR program during regularly scheduled hemodialysis sessions. So we were looking at primary few questions here. Do patients get motion sickness, like symptoms from full immersive VR? So using fully immersive VR, you may experience some motion-like symptoms such as fatigue, nausea, headaches, dizziness, very much similar to the symptoms that patients already have during hemodialysis. So we also wanted to look at, does the fully immersive VR exacerbate any of those symptoms that patients have during dialysis? And then we also want to look at if fully immersive VR is patient-friendly. And lastly, if we can actually implement a virtual reality program successfully in a hemodialysis clinic. So our methods, inclusion exclusion criteria, very standard for trials here in the United States. One of the biggest ones here, though, being that it was fully immersive VR, is a history of epilepsy, seizures, or vertigo. So VR, full immersive VR can kind of initiate those episodes. So we excluded that. We had 20 patients go through the program from start to finish, and then kind of just highlighted here, the VR familiarity, that's individuals, so 65% of the individuals knew what full immersive VR was. They had an understanding of what it was, versus 30% actually had some type of experience prior to the study that they were exposed to full immersive VR. So we used an Oculus Rift to immerse the participants in the VR world. Again, it's utilized a kind of over their eyes headset. The exposure was during hemodialysis treatment sessions, and the exposure was on 2 separate days, but in consecutive treatment days. So for like a Monday or a Wednesday or a Tuesday, Thursday schedule. Our program centered around mindfulness, and it used, we kind of had 2 portions, 2 parts of this mindfulness program. C1, we used didactic material on the benefits of mindfulness, so how patients can utilize mindfulness in their everyday life. And then scene 2 is actually where they practice some breathing exercises. So each scene was about 10 to 12 minutes, lasting for about a total of 20 to 25 minutes. Some outcomes we had here, the primary outcome was the simulator sickness questionnaire. This is 15 items, symptoms that patients can kind of check their severity of at the given time point. So the questionnaire or the symptoms are very much related to what, again, the patients would be experiencing during hemodialysis. So we gave this questionnaire right before they were exposed to the VR program, and right afterwards, right after they took the headsets off. Then we also had secondary outcomes of the eye presence questionnaire. This questionnaire assesses the individual's level of presence or immersion into the VR world. So how much they felt that the VR world was real, that they felt that they were actually in that VR space. And we gave this questionnaire directly after they finished that symptom simulator sickness questionnaire post VR exposure on both days. Then the system usability scale was used to assess kind of patient friendly, how they interacted with the space, with the VR equipment, if it was usable, if it was patient friendly, and we gave that at post intervention. And then we also had some qualitative feedback questions from the patients and clinical staff to assess whether or not this was in fact feasible to conduct in a clinic. So we found out that a single 25-minute VR-based mindfulness program significantly lowered their symptoms. So as you can see here on the left-hand side, the 15 or so different symptoms that are both associated with hemodialysis and also with motion sickness through full immersive VR. So we found a significant reduction in exposure 1, so day 1, in their total symptom score. And then for day 2, while it wasn't significant, we did see a reduction in their symptoms as well. So our hypothesis of that we thought it may exacerbate the symptoms was actually false, and that we saw a reduction in their symptoms. So then we also looked at the eye presence questionnaire and the system usability square, and we determined that it was patient friendly. The individuals had high levels of immersion, so this is scale goes to 7 on the IPAC score, and sorry, there we go. The involvement and realism was a little bit low because they actually didn't have much interactions with our VR space the way we had it modeled. It was more of just kind of sitting back in a third person situation. The total score of usability, that's out of 100, so pretty high on the usability. So we determined that it was pretty patient friendly. The main thing here was that individuals didn't really like some of the graphics that we had, but other than that, they seemed to enjoy it a lot. And then as far as the implementing in a hemodialysis clinic, all the staff pretty much said that it didn't disturb, excuse me, the care that they were giving to the patients. None of the staff felt that we were in their way at all, that they could still communicate with the patients even though that they had a VR headset over their eyes. So we determined that it was successful and being implemented in a hemodialysis clinic. So in conclusion, a 25-minute fully immersive VR is safe for hemodialysis to experience while receiving hemodialysis treatments. It is patient friendly. It did not cause any disruptions in the clinical staff or for the care of the patients, and it may be a suitable alternative mode for intervention delivery. Some limitations: we didn't have a control group or a control day, so we don't know specifically if, in fact, those reductions in symptoms was from the VR program. And then we also had a very small sample size, only 20 participants. Some future research, we're writing a grant right now to include a control group and increase the sample size. And then we're also looking at utilizing full immersive VR in educational programs, such as grocery shopping tours for the patients, and implementing an exercise intervention utilizing full immersive VR. So thank you very much, and I'll take any questions.
Thank you very much, Brad, for your presentation. So let's see if we're going to receive any questions. We have a question, Brad, from Noemi. So, Brad, you mentioned that qualitative feasibility outcomes from clinic staff and patients. What were these outcomes? For example, recruitment rate or?
Yeah, so mainly we looked at kind of the aspects of the program, kind of patient friendly. Did you like the graphics? Were you able to navigate through the VR world without any trouble? So it was more of that versus not necessarily more of the recruitment aspect of it. It was more of the programming because we're looking again to doing a bigger study and kind of focusing more on the patient's kind of experience in the VR world.
Thank you for your answer, Brad. Could you tell us about the cost of implementing interventions like that?
The cost?
Yeah, the cost, please.
So we have actually a VR lab on campus where they were just starting out and actually did this work pro bono. So we were very fortunate to get, they created this VR world for free, which was great. But we did have some kind of hiccups in there a little bit where we had to outsource to a actual VR company, and it was quite expensive. So that's why we're writing a larger grant to do a kind of a bigger study, because with the VR, that is just the kind of the creation of the world is quite expensive. But I don't know the detailed specifics. I know we got a small grant of about $8,000 and that covered it. So, but just barely. So it can be very expensive.
Okay, got it. Thank you, Brett. We have another question from Annabelle. What did the participants think about the content of mindfulness?
So they kind of, they really enjoyed the meditation portion of it, the second part of the program. In fact, about maybe 40% of the participants fell asleep while they were doing it. So they all enjoyed it a lot. They found it very relaxing. But the only thing, so we did the exact same protocol or program both days, and they thought it was a little boring. So we know to kind of either switch things up a little bit or gamify it a little bit more for the next iteration. But they did enjoy it. They enjoyed also the fact that the way that we had the didactic material on the educational component on how to give them tips to kind of utilize the mindfulness training kind of in their everyday life.
Perfect. Thank you, Brett. And the last question from Noemi. You mentioned 8 staff members. What were their professions?
So there was various staff members. They were mostly technicians and nurses. There was one, I believe one of them was a social worker as well, that had kind of more of a interaction with the patients, but no nephrologist, because honestly, we didn't have any nephrologists come in the doors while we were during the intervention. So we didn't get any feedback from the nephrologist.
Okay, thank you. And last question from Sadia. Is there a classification to the IPQ score?
So it's based out of 100 is the max score, and higher scores meaning, so that's actually for the system usability scales, the 82, 75. The IPQ score we had about a 4 or 5 on the scale, and that's out of 7. And to be honest, there is actually no known research on the kind of level, but from talking with some other researchers in the field, has a very good score of about a 4 or 5 on a level of 7. Having a 7 is very difficult, if not impossible, to have full immersion where a person feels like they're 100% in that VR world. They still feel like they're kind of present in the kind of real-time world.
Okay, perfect. Thank you very much for your presentation, Brett.
Sure, thank you.
Yeah, thank you very much, Brett. We're gonna have a next presenter. Again, Brett from Australia. Brett Tarca, he's an exercise physiologist and a PhD student at the University of South Australia. Brett completed his master's by research in February 2020, which was also working with people receiving peritoneal dialysis before commencing his PhD in April 2020. Brett's PhD research focus is on fatigue, mood, and physical function and physical activity for people receiving peritoneal dialysis. And with his presentation today, he is going to talk about physical function for people receiving peritoneal dialysis. So Brett, whenever you're ready.
Thank you, Naomi. Beautiful. Good morning, good afternoon, good evening, wherever you happen to be. I just want to start by thanking the GREX for this wonderful opportunity to present some of the fabulous work that we're doing here at the University of South Australia surrounding the physical function for patients receiving peritoneal dialysis. I'm just going to refer to them as PD for the remainder of this presentation. So why physical function, and why is it important to people on PD? Well, last year there was an international consensus meeting that took place that involved people receiving PD and their caregivers. And the aim of this meeting was to identify research priorities. And what the patients and the caregivers identified was the ability to do usual activities was the third highest priority out of 36. And it's not surprising, given that the literature reports that patients receiving PD often have a physical function around 50 to 70% of age and gender matched peers. So the ability to do usual activities, this is synonymous with the definition of physical function. What we know about physical function is in part made up of determinants that are modifiable, including aerobic capacity, strength, activity, and sedentary behavior. We also know that patients that tend to have higher physical function levels tend to have a higher quality of life, lower morbidity, and lower mortality. But from the perspective of PD patients, if we're going to intervene and design exercise programs that are optimally designed for functional outcomes, we need to know which one of these modifiable factors should we be targeting and which one's more or less going to give us the best bang for buck. So the first step in this process was to establish what we do and don't know. And we did this through a systematic review. So the question we were asking was, what are the modifiable physical factors associated with physical function for people receiving dialysis? So the review was open to both dialysis modalities. A modifiable factor was deemed one that was potentially modifiable by exercise intervention, such as the examples you can see surrounding physical function. Our outcome definition was function, and we define this as ICF measures of activity or participation. And this is just a few assessment examples for each respective category. It's also very important to note that to be included in the final review, it must have a statistic that can be classified by degree of strength. So we're talking about correlation coefficients or effect size values, statistics like that. So the search strategy was quite robust. It yielded quite a few studies to begin with, which were able to find 23 that met the inclusion criteria for the final review. The majority being cross-sectional or case control, and the overwhelming majority was investigating hemodialysis populations. We were able to extrapolate 126 associations, 100 of which were found to be significant. So this is the key finding from all those studies. That number you can see below each category, that's the number of significant associations for each respective category. We also classify the strength of association according to Cohen's stipulation, which is either weak, moderate, or strong. Physical activity, as you can see, had the overwhelming majority of these associations. And it's also important to note that from these 52, 48 of these were either moderate or strong. So it allowed us to conclude that physical activity had a consistently moderately strong relationship with function, as in those who had higher activity levels tended to be of a higher function. The remaining 4 categories, unfortunately, didn't produce such consistent results. And as such, we weren't able to draw any conclusions about muscle size, mass, strength, and sedentary behavior. Some other key findings from this review was that we weren't able to identify any cardiorespiratory fitness link with function, which we were quite surprised with. The PD population made up only 4 of the included 23 studies, so very under-researched. And there was very much limited longitudinal data. This made up only 2 of the final 23 studies. And if you'd like to read more about this, this has been published in the Journal of Physical Activity and Health. Now, I'm making quite a segue here into abdominal strength, and you're probably wondering why. It's not really surprising when you think about it, because abdominal muscles play such a pivotal role in many daily life processes and functions. And if you know, obviously for people receiving PD, they do have a catheter placed in the stomach. This involves a surgical incision through the abdominal wall. And what we don't know is, does this lead to impairment? And again, more often, does having fluid permanently in the cavity or for a prolonged period of time result in any abdominal impairment? So one thing that we don't know is what's going on with the abdominal muscles for people receiving PD, and probably more importantly, how can we assess them? Unfortunately, the literature doesn't have many suitable and/or valid tests to assess abdominal strength. So we went about validating an assessment, a tool and a protocol. So a study that was in my master's was to investigate the validity and reliability of handheld dynamometry, which is that little gadget you can see in the middle. against isokinetic dynamometry, which is considered the gold standard for muscle testing. It is worth noting we had to do this in an apparently healthy population, as this was the first step before getting clinical validation. The protocol we were looking to validate is what you can see in the bottom right-hand corner there, where I've got my colleague Chris on a 25-degree bed wedge, putting him into 25 degrees of trunk flexion with the handheld dynamometer sitting on his sternum. The isokinetic dynamometry position, which is in the top right-hand picture, as you can see, is quite different. And you're probably wondering, why aren't these pictures the same? Very simply, we weren't able to replicate the bottom right-hand setup with the isokinetic dynamometer arm. However, we did conclude that this difference was negligible and shouldn't influence the results greatly. So what we found from our testing procedure in this apparently healthy population was that the intra-class correlation coefficients for validity was good, and the reliability was also good. The graph you can see in the middle is a Bland-Altman plot, and what this was designed to do was to measure any proportional bias, to which there was none. So we were able to conclude that the device and the setup was both valid and reliable. However, the clinical validity was still in question, so the ability to predict unfavorable outcomes such as functional decline. So we were really set now to obviously test this out with patients receiving PD. If you'd like to read more about it, this has been published in the Journal of Sport Rehabilitation. So back to physical function now, and which one of these modifiable factors are we going to target for patients on PD? So our aim was to figure out which objectively measured modifiable physical assessment category is associated or predicts physical function. So what in short, we want to know, is it muscular strength, is it cardiovascular capacity, or is it physical activity and sedentary behavior that is the strongest predictor of their physical function? For muscular strength, we're running 4 tests. We're using handheld dynamometry to assess quadricep strength, bicep strength, and our now validated abdominal strength. We use a hand grip dynamometer as well to assess hand grip strength. We're using the 6-minute walk test for cardiovascular capacity, and we use accelerometry and inclinometry, excuse me, to assess physical activity and sedentary behavior. It's worth noting that this, to our knowledge, is the first study to use inclinometry to capture objective sedentary behavior in this population. Our functional outcomes are the short physical performance battery and the SF-36 physical functioning scale. We're running it over a 12-month period with measurements taken at 3 time points. So from a recruitment point of view, we're progressing quite nicely, given we do have a limited sample size here in South Australia. And it's worth noting as well that I am performing some of these assessments at participants' homes. This was done to obviously open up, give us further reach. So participants have the option at completing at their home or at a routine visit at the local dialysis clinic. So just a little bit of baseline descriptive statistics. We haven't gone too far in with the statistical stats yet, as we haven't got enough longitudinal data, but just a few things I'd like to highlight. The 6-minute walk test distance, this is actually quite low. This is so this average of 3:14. I know that normative data should always be used with caution, and it is quite varied, but that is very low based on the mean age of just nearly 64 years. It's also quite low to other PD cohorts that have been reported in the literature. It's worth mentioning that our abdominal strength test assessment, I would deem that to be acceptable. So 57 of the 58 have completed this assessment with no adverse event being reported. So we do conclude at this point in time that it has been accepted and it is suitable. The sedentary time that we're capturing through the inclinometry, there was a survey done in 2016 about older Australian adults, and this number of 10.6 hours is about 3.5 hours more than the average older Australian adult. So it's quite significantly more, which is obviously detrimental if they've got more sitting time. And moderate activity, it's advocated here in Australia that generally speaking, people should be completing 30 minutes of moderate activity a day. And so the patients receiving PD are obviously not completing as much as is recommended. So recruitment is ongoing. It's a very exciting project. It's got a lot of firsts in it. So obviously the first to assess abdominal strength in this population, as well as capture objective sedentary behavior with inclinometry. Also, it's the first one to have a robust testing battery that includes multiple modifiable factors. I'm about to launch into my next investigation, which is going to be looking at fatigue, mood, and physical activity. And this is going to be commencing very soon. I just want to thank my supervisors, Katia, Tom, Paul, Shilpa and Anthony, who have been just outstanding so far. The Central Northern Adelaide Renal Transportation Service Clinical Research Group, who are collaborating on all these projects and again have just been wonderfully supportive. And very importantly, the Hampstead Dialysis Home Therapies units, who have just been unbelievable in terms of getting a lot of patient supports and a little bit of excitement about exercise, which I'm sure most of you know can be quite a challenge. Thank you very much. I'll be taking any of your questions now.
Thank you very much, Brad, for your presentation. Congratulations, just awesome. We have a question from Thomas Wilkinson. Brad, do you have any data on the inter-rater reliability on your handheld device?
No, I don't. No, we only did the intra, so it was only done by me. So short answer is no. I don't have any inter-rater reliability data to present. Sorry.
Okay. Do you think it depends on the ability of the tester to be able to withstand the strength of the person being tested, or do you need to be strong bloke to be able to keep the device steady?
I've got a very limited sample size to really operate on, and to be fair, they haven't really overpowered me. I'm the one doing the testing. It's a good point. I can't answer it because I haven't really come across too many highly athletic people with very, very strong abdominals, particularly in this population. So I'm going to say that the jury's still out on that one. We need further research. It's very, very preliminary in terms of where that validation sits right now.
Okay, thank you for your question. We have a question from Krista Stewart. What is the test used for sedentary time, and how is it performed?
Yep, so it's using an inclinometer. So an inclinometer is a matchbox about the size of a matchbox. It sits on the participant's mid-thigh. And so what the inclinometer does is it actually measures the person's body position relative to the ground. So if you think about that upper thigh, that's how it's able to distinguish between sitting, lying, and standing. And yet that's how we're capturing objective information. Typically, people have used accelerometry, which doesn't quite capture how much time spent sitting. So it can't, capture that because it doesn't know the body position. It just knows the intensity that the person is working at. So that's how we're using inclinometry.
Thank you for your question, for your answer, Brad. We have a question for your supervisor, Paul Bennett. How does the PD abdominal strength compare to other 6-year-old populations?
Yep, so I did have a look at this last night in preparation for a question like this. So not that I promise you, Paul didn't give me a leg up here. So there's obviously, because the test is so new, it's very hard, there's no normative data for it and it's very hard to compare. However, I did go back and look at that validation study and looked at people that were over the age of 50. So there was 11 people aged over 50 and the average strength of those people was at 13 kilos. So how does it compare? On average, the apparently healthy population's abdominal strength was pretty much double that of PD patients, which I find interesting, but we do need to test that clinical validity before we can really draw any comparisons with that population.
Okay, thank you very much, Brad. One more question from Pedro Martins. What do you think abdominal strength data adds to other strength measures as hand grip strength?
Can you elaborate a bit more? I'm not quite sure what he's asking.
Yeah, probably Pedro is asking about what do we get from testing abdominal strength than we get from measuring hand grip and leg muscle strength.
Yep. So the benefit of measuring abdominal strength is, well, for one, we just don't know what's happening with that strength, but two, we still don't know if it's if it does have a link with physical function. So it's a little bit preliminary to really conclusively answer that question, because we are this is one of the first tests to actually assess the clinical validity and utility of the tool. So in terms of what we can get from it, it's a bit too early to know. Hopefully when I get more data, and particularly a lot more longitudinal data, we're going to start to see, is there impairment, and does this impairment correlate with a functional outcome? So we're going to get to see, is the abdominal strength playing a pivotal role in the functional status of this population?
Okay, thank you. And Pedro is asking also about, is there any association between the abdominal strength and PD related outcomes, for example, peritonitis?
Not yet. No, no. This is one of the. So this is without giving too much away. It's something that does interest me. It's been often hypothesized that increased diaphragmatic pressure can lead to things such as peritonitis, hernias, or even catheter leaks. These not quite substantiated yet whether the strength is related to this, but it's definitely something that we're interested in, and watch this space.
Okay, thank you very much for your answer, Brad. So thank you for your presentation, and
Thank you.
let's go to the next one.
Yeah, thank you very much, Brett. So the next presenter is Tom O'Sullivan from Leicester. So Tom is a PhD student from the University of Leicester in the UK. During his project, he's investigating the mechanisms of skeletal muscle wasting in patients with non-dialysis CKD, with a particular focus on inflammation. So Tom, whenever you're ready.
Okay. Okay, hopefully you can all see my screen. Hi everyone. So yeah, I'm Tom from Leicester, and I'm going to be presenting some data using a primary skeletal muscle cell model from CKD patients. So we know that muscle wasting is a serious issue in CKD. It shows associations with mortality, reduced physical function, and reduced quality of life. However, despite these clear clinical implications, our understanding of why this happens is incomplete. So research to date uses a combination of muscle biopsies from patients or animal models of CKD or immortalized cell lines. And although a great deal of progress has been made in this using these approaches, there are limitations. So for example, biopsies only provide a single snapshot of the situation with limited scope for experimental manipulation. This is afforded by CKD models, animal models, and immortalized cell lines, but then these are limited in terms of their translation to human physiology. So essentially, we need better tools to study this. So research in other areas in clinical populations where we see muscle wasting are more increasingly using primary skeletal muscle cell models, where you isolate muscle cells from tissue and grow them in culture, which provides an in vitro model of myogenesis. Now, myogenesis is a process whereby satellite cells provide new myonuclei to mature myofibers. So satellite cells are essentially skeletal muscle stem cells. They reside in a dormant state in resting skeletal muscle in response to stimuli such as exercise or damage that are activated to produce proliferating myoblasts, which then differentiate and fuse to the mature fibers. Now, this process is regulated by the sequential expression of a number of transcription factors, PAC7, MIF5, MyoD, and myogenin. Not only do they regulate this process, but they also provide molecular markers of progression through myogenesis. So PAC7 we see in quiescent and activated satellite cells, and then reduces during differentiation. Whereas MIF5 and MyoD are found in activated and proliferating myoblasts as well. MIF5 reduces during differentiation, whereas MyoD continues, where we also see an increase in myogenin. Other research in populations such as diabetes and also COPD show that characteristics seen in vivo from donors of primary muscle cells are then retained by the cells themselves. For example, cells taken from insulin-resistant individual are then insulin-resistant when we grow in culture. However, as there's a lack of human primary cell work in CKD, this has never been done before. We don't know if this is also the case in this population. So with this in mind, the aims of this work was to firstly establish a human primary cell culture model of muscle in CKD, and then use this model to determine if there was a uremic phenotype retained by these primary muscle cells, i.e. do we see characteristics or hallmarks of muscle wasting that we see in vivo then retained in these cells. So the way we did this was to take biopsies from non-dialysis dependent CKD patients isolate the satellite cells and then grow them in culture for experimentation. Samples were taken from 4 patients and 4 age and sex matched controls. There was a significantly reduced GFR in the patient group, but a similar Desmond index in the myoblast cultures isolated. So this is an overview of the experiments itself. So myoblasts were seeded into growth media with a high serum content to stimulate proliferation, which was assessed over 4 days, 4 consecutive days using the Alamar Blue assay. On day 3, myoblasts were harvested for mRNA analysis before differentiation was induced on day 4 by replacing media with a differentiation media which had a lower content of serum. These cells were then grown for a further 7 days, after which morphology, mRNA, and protein analysis was performed. So there's quite a lot of results to get through, so I'm gonna crack on. Here we see an immunofluorescent image of the myoblasts, and they presented as mononucleated cells positive for the myogenic protein desmin. Hopefully, as you can see from this right field image, the cell density increased over time, showing that these myoblasts progressively proliferated over time in culture. However, when they were induced to differentiate by reducing the serum content, we see that these mononucleated cells fuse to form multinucleated myotubes. So that indicates that they successfully differentiated. I also wanted to look at the gene expression profile of these cells to see if it replicated what we would expect of myogenesis. So this table shows the fold change in each marker in myotubes compared to myoblasts. And there was a reduction in PAX7 gene expression, although this was only significant in healthy control derived cells. There was no significant difference in MyoD, whereas MIF5 was reduced following differentiation, whereas the opposite was seen for myogenin, which significantly increased, which replicated what we would expect of in vivo myogenesis. I also looked at myosin heavy chains. Now these are expressed by mature skeletal muscle, so we would expect to see an increase in myotubes following differentiation, and that was indeed what we found, with significant increases in all myosin heavy chains I looked at in both donor groups. So basically what this shows here is that the model replicated myogenesis and therefore appears to be a good model for mechanistic research. So moving on to the comparison between donor groups, in terms of myoblast proliferation, it increased significantly over time in culture in both groups, but there was no difference between groups. In terms of differentiation, following 7 days, there was no difference in the diameter of myotubes or the fusion index, which indicates the number of myonuclei which are incorporated into myotubes, showing that cells from health controls and CKD patients had a comparable capacity to differentiate. However, I did see differences in myogenic regulatory factor gene expression, which is generally higher in CKD-derived cells. So PAX7 and MIF5 expression was higher in CKD myoblasts on the left of the figures here, and also myotubes. And MyoD gene expression was also higher in CKD myoblasts. However, there was no difference in myogenin gene expression in either myoblasts or myotubes, or in any of the myosin heavy chains, which I haven't shown on this slide, but there were no differences. I also looked at some inflammatory markers and saw an increased gene expression of IL-6 in CKD-derived myotubes, but no differences in TNF-alpha or myostatin. Now, I also looked at markers of protein degradation, MRF1 and MAFBOX, but there was again no significant difference, although there was a trend for increased MAFBOX expression in CKD-derived myotubes with a large effect size. Finally, I looked at AKT phosphorylation. Now, AKT is a protein which is important for maintenance of muscle mass, and has the phosphorylation of this protein has been shown to be reduced in biopsy samples from CKD patients. However, this doesn't appear to be something which is retained in primary myotubes and culture, with no difference in AKT phosphorylation between groups. Okay, to summarize this data, regarding the first aim of establishing a human primary cell culture model of skeletal muscle and CKD, I demonstrated that myoblasts proliferated progressively over time in growth media with high expression of PAC7 and the proliferative marker MIF5. When these cells were induced to differentiate, they fused to form multinucleated myotubes, and the expression of differentiation markers, myogenin and myosin heavy chain, increased. Regarding if there was a uremic phenotype that was retained by primary muscle cells in vitro, I saw no difference in myoblast proliferation or myotube differentiation, and also no difference in AKT phosphorylation. There was a trend for increased MAFBox mRNA and IL-6 in CKD-derived myotubes, and generally increased myogenic regulatory factor gene expression in CKD-derived cells. So what does this mean? I suppose it shows that this model of human primary skeletal muscle cells is suitable for mechanistic research into muscle wasting in CKD. However, regarding the uremic phenotype, I don't think there's strong evidence to show that this, well, this evidence doesn't indicate that this was the case. However, this is caveated by the fact that only a limited number of outcomes were assessed. So there may be differences in ones which I haven't considered, for example, mitochondrial function or protein synthesis or protein degradation. Also, a lot of this data relies on gene expression, which doesn't infer changes or differences at the protein level. So I think further characterization is needed of the model to fully determine if there is indeed a uremic phenotype or not. And if there isn't, this may indicate that it is something within the CKD environment which is driving muscle wasting in this population. But once these cells are removed from that environment, they then sort of behave similarly to healthy control cells, which is something which we've gone on to look at. So thank you very much for your attention. And is there any questions?
Thank you very much for your presentation, Tom. Very nice basic science study. Well done. We have a question from Daniel Kirkman. How much do you think the in vivo uremic and systemic inflammatory environment in CKD contributes to your markers? And can you replicate this environment in your culture model?
So how much do you think they will? So yes, we can. Well, we can try to replicate this in my model. So what I've then gone on to do is start culturing these cells in serum, which is isolated from patients and also controls, and then look at the same outcomes. So yeah, I did see some differences. So I found that human serum from CKD patients increased myoblast proliferation and also reduced the diameter of myotubes, either ones derived from healthy controls or CKD patients. How much this is due to the CKD environment is less clear, because I saw similar but reduced changes in response to healthy control serum as well. So it might be something in the model itself that requires optimization until we fully determine if it's something specific to the uremic serum or just human serum in general. But the evidence, or the preliminary evidence I have so far, suggests that there is something in CKD serum which was stimulating these responses. Again, what that is, I'm not entirely sure yet, because just by adding serum, you're adding a whole plethora of different factors, a lot of which are affected by CKD. So it would then be a case of looking within that serum to find the source of any differences or any effects that we see.
Okay, thank you for your answer, Tom. Let's see if we have more questions. Okay, you have a question from Courtney. Great presentation. How healthy are your healthy controls? Are they healthy or non-CKD?
So they're non-CKD first and foremost, and they're generally healthy. We exclude anyone with with diabetes or generally any condition which could negatively impact skeletal muscle mass. So certain types of cancer. So yeah, they're generally healthy individuals, and they are recruited from theatre. So they are going in to have a procedure such as the removal of a benign lipoma or growth from their leg, and once the surgeon's in there, then we ask them if they can also take a sample that we can use. But yeah, in answer to the question, yes, they are generally healthy, not just non-CKD patients.
Okay, thank you, Tom.
Thank you very much, Tom. Just before everybody goes, I would like to share my screen and talk a little bit about our future webinars. So, sorry. So our next webinar is going to happen on the 19th of November, and abstract deadline for it is the 5th of November. If you would like to register for a GREX or GREX membership, you can do that on our website, and it is free and quite easy to do. If you're interested in submitting an abstract, just please find the link in this slide, or you can contact any of us via Twitter or the website. On behalf of the whole organizing committee, I would like to say thank you for coming today, and thank you very much, of course, to the presenters, and I hope you all enjoyed it. So thank you very much. And now it's 15 minutes of questions and answers to all of the presenters, or about GREX or GREX in general. So thank you. Okay, I actually have a question to Brett Tarca. Not sure if he's here. You there? Okay. So you mentioned that you are, so you're still recruiting for your study.
Yes, correct.
Right. So here in the UK, we can't really, well, we are just trying to do studies, but we can't really do sort of like face-to-face patient studies at the moment. So I know the situation is very different in Australia, but I wondered how like COVID affected your recruitment rate, or have you noticed that it had an effect?
I've been very, very lucky. The situation here, when it was at its height, it's actually, I've been incredibly lucky, really. So obviously there's 6 months between testing points, and when the height of the COVID restrictions hit, which was in April and May, this was actually a down period for me, where people kind of fell in between their testing time points. So I missed 4 assessment periods due to those restrictions. But I have been very fortunate, touch wood, that I haven't been affected too much. So short answer is very lucky, but no, obviously very, very sensitive to the situation, and it can obviously, as we've seen, it can change quite rapidly. Yeah.
Have you seen that patients generally request more home visits? Do they, so you do the outcome test at home, so do they generally prefer you to go to their homes, or do they come to the hospital?
The majority have been at participants' homes. So one of the reasons why, in part, why PD cohorts are so poorly represented in the literature is because they're so geographically spread. Obviously living in different locations. So Adelaide, probably not, there's probably not many who are aware of the actual how big Adelaide is, but it's not too bad compared to other major cities. So it hasn't been too bad going to people's homes. And yes, they have preferred it, probably marginally more. It's almost about a 60-40 split between home to clinic visits.
Okay, right. I'm just jealous of you.
Oh.
We all are, knowing me. We all are.
Yeah.
Okay, that's it.
Okay.
Thank you very much, guys, for your time. Hope to see you in November.
Yeah, see you soon. Thank you very much.
Bye-bye.
Congrats. Bye.